So if we go back to October 1951, there was a 31-year-old woman named Henrietta Lacks, and she tragically passed away from this really highly aggressive form of cervical cancer. But, well, a few months before her death, doctors actually took a small tissue sample from her tumor. And when researchers put that sample into a culture, something completely unprecedented happened.
Right, because for the very first time in medical history, these human cells didn't just, die after a few days in a lab dish. They actually survived.
Whoa.
Yeah, they kept dividing completely unchecked. And those specific cells, which they dubbed HeLa cells, just taking the first two letters of her first and last name, they basically became the first immortal human cell line.
Which is just, I mean, it's an incredibly heavy, almost science fiction reality to wrap your head around because those original cells taken over 70 years ago are literally still alive today.
Yeah, they are.
They've been multiplying reliably for decades. I mean, they helped researchers develop the first polio vaccine. They've been used to study the effects of radiation. And this blows my mind. They were literally sent up into space to see how human tissue reacts to zero gravity.
It's amazing. They completely overturned our understanding of biology. Because,
Yeah.
well, if you want to understand how any living thing functions, whether that's a fungus, a redwood tree, or, the human being listening to this audio right now, you have to understand the fundamental building block of life. You have to understand the cell.
Okay, let's unpack this. Because our mission for this deep dive is to explore that exact invisible architecture. We are going straight into the microscopic metropolis today.
I love that description.
Thanks. Yeah, we need to figure out the tools that even allow us to see this hidden universe and how our own cellular factories operate, the mechanisms behind the bacterial invaders we constantly fight off, and like how billions of these tiny units cooperate to build you. But before we get to immortal cells going into space, how did we even figure out we were made of these invisible blocks in the first place?
Well, for the vast majority of human history, we were just entirely oblivious. The microscopic world was basically dark matter to us.
Right.
The curtain didn't really begin to lift until 1665. There was this English natural philosopher named Robert Hooke, and he designed this early optical microscope. So he put a thin shaving of cork under the lens and noticed it was made up of these tiny, empty, regular compartments.
And he called them cells, right?
Exactly. He called them cells because they reminded him of the austere, cramped quarters that monks lived in at a monastery.
That is a surprisingly poetic origin story for the word. But, I mean, a slice of dead cork is one thing.
Yeah.
When did we actually see something moving?
That happened about a decade later with Anton van Leeuwenhoek.
Oh.
He was actually a Dutch draper, and he was meticulously grinding his own lenses just to inspect the thread count of his fabrics.
Wait, really? Just for fabric?
Just for fabrics.
Eh?
But out of curiosity, he decided to look at a drop of pond water. And through his highly polished lenses, he discovered an entire bustling ecosystem of single-celled organisms just darting around.
Oh, wow.
Yeah, and he affectionately called them animalcules.
Animalcules. That is objectively brilliant. I love that. But jumping from seeing tiny creatures in pond water to realizing that literally everything is made of cells feels like a pretty massive leap.
Well, it was a massive leap, and it took until 1839 for Matthias Schleiden and Theodor Schwann to formalize it into cell theory.
Right.
They proposed that cells are the fundamental unit of structure and organization in all living organisms.
Yeah.
But the roadblock they hit was technological. They were limited by the actual physical properties of light.
Right, because there is a hard limit to how far you can zoom in with a traditional glass lens. I think a lot of people, myself included, kind of assume you can just stack more and more lenses to get closer, but that's confusing magnification with resolution.
Exactly. That is the crucial distinction in microscopy. Magnification just tells you, how much bigger an image appears. But resolution is the ability to distinguish two separate points as distinct objects.
Okay, so how does that limit us?
Well, think of a standard light microscope. It uses visible light, and it can resolve down to about 0.2 micrometers. If two tiny structures inside a cell are closer together than that 0.2 micrometers, the light waves essentially just blur them together into a single blob.
So even if you magnify that blob like 1,500 times, you just end up looking at a giant blurry blob?
Precisely.
Yeah.
Now, light microscopes are still fantastic for certain things. You can look at living, breathing organisms. You can actually watch an amoeba hunt. But because biological tissue is mostly water and pretty transparent, you often have to use specific chemical stains to even see the structures. Hematoxylin is a really common one.
What does that do?
It binds to the nucleus of a cell and turns it a deep purple or blue. Or you might use iodine to react with starch in plant cells turning those areas blue-black.
Got it. But if we are stuck at that a zero point 0.2 micrometer resolution limit, how did we ever get the blueprints of the internal cellular factories? Because we obviously know what the inside of a cell looks like today in like staggering detail.
We do. And to crack that, we basically had to abandon light entirely. In the mid-20th century, researchers developed the electron microscope.
Okay, so how is that different?
Instead of bouncing visible light off a specimen, it fires a highly focused beam of electrons. And because the wavelength of an electron is just monumentally shorter than the wavelength of visible light, the resolving power just skyrockets.
Wow.
We're talking about resolving details down to 0.1 nanometers and magnifying objects up to 500,000 times.
That is unbelievable. At that scale, an average human being would look, what, over 800 kilometers tall?
Yeah, easily. It opened up a completely new frontier.
Yeah.
So you have transmission electron microscopes, or TEMs, and those pass electrons through a specimen to give us hyper-detailed 2D internal cross-sections.
Right.
Meanwhile, scanning electron microscopes, or SEMs, bounce electrons off the surface to generate those incredibly dramatic, highly textured 3D images of like pollen grains or insect dyes.
I'm hung up on the mechanics of this, though, because if you are firing a beam of electrons, wouldn't they just smash into the oxygen and nitrogen molecules in the air and scatter everywhere?
They absolutely would, and that is the massive trade-off of electron microscopy. To keep the electron beam perfectly straight, the entire inside of the microscope has to be a flawless vacuum.
Which means any cell you put in there.
Must be completely 100% dead.
Oh, wow.
Yeah, all the water has to be chemically extracted. The specimen is embedded in hard resin, sliced incredibly thin, and then to give the electrons something dense to bounce off of so we can actually see a contrasting image, the tissue is stained with heavy metals. We use lead and uranium.
So I always try to visualize it like this. A light microscope is like looking at a live, bustling city map. You can see the traffic flowing, you can see where the parks are, it's dynamic. But an electron microscope is like Google Street View. You can zoom all the way down and read the serial number on a fire hydrant. But to get that view, you had to freeze the entire city in time, kill every living thing in it, suck out all the water, and coat the skyscrapers in radioactive heavy metals.
That is a perfect analogy. It's a brutal chemical preparation. And because of that brutal preparation, researchers constantly battle with something called artifacts.
Artifacts, like historical objects.
No, in this context, artifacts are physical distortions, like weird clumps or fractures that show up in the final image. They aren't actually part of the natural cell.
Oh, I see.
Yeah, they're just damage caused by the heavy metal staining in the vacuum.
Yeah.
So the real challenge of early cell biology was figuring out what was a real cellular structure and what was just a preservation artifact.
Okay, so assuming we account for the artifacts, what does that lead-coated Google Street View actually reveal when we look inside our own eukaryotic cells?
Because the human body isn't just a bag of a cytoplasm, right?
Hey, far from it.
Yeah.
When you look at a eukaryotic cell, which is the complex type of cell that makes up plants, animals, and humans, you find a highly organized, heavily compartmentalized metropolis.
Okay.
The bulk of the cell is the cytoplasm, which is a jelly-like matrix holding everything together. And floating inside that, you have the nucleus. Together, the cytoplasm and nucleus make up what we call the protoplasm.
The nucleus is the massive control center, right? It's usually the biggest thing you see under the microscope.
Exactly. It's surrounded by a double membrane full of selective pores. Inside, the nucleus holds chromatin, which is your raw DNA, tightly spooled around protective proteins.
It also contains an extra dense region called the nucleolus, which has a very specific job manufacturing ribosomes.
I want to hold on ribosomes for a second, but if the nucleus is just sitting there holding the blueprints, how does the cell physically divide when it's time to replicate?
That's where centrioles come in. Just outside the nucleus, eukaryotic cells have these structures called centrioles. They look like tiny bundles of microscopic pipes, specifically 9 sets of tubules.
Okay.
When it's time for the cell to divide, these centrioles migrate to opposite ends of the cell and cast out a massive net of microtubules. This is called the spindle, and it literally grabs the chromosomes and physically pulls them apart into 2 new cells.
Okay. Pulling chromosomes apart requires serious mechanical effort. Operating an entire cellular metropolis requires massive amounts of energy. So where's the power grid?
That would be the mitochondria.
Ah, the powerhouse of the cell.
Exactly. They are tiny, rod-shaped organelles scattered throughout the cytoplasm. Their entire purpose is cellular respiration. So they take glucose and oxygen and churn out ATP, which is the chemical energy currency of your body.
What's always fascinated me about mitochondria is their physical architecture. Because they have an outer membrane, but the inner membrane is wrinkled and folded back on itself dozens of times.
Yes, and those deep folds are called cristae. See, in biology, form always follows function. Whenever you see a heavily folded membrane, the evolutionary goal is to maximize surface area within a tiny volume.
Oh, that makes sense.
The entire inner surface of those cristae is studded with the enzymes necessary to produce ATP. So if a specific cell needs massive amounts of energy, like, say, a muscle cell in your thigh or a heavy processing liver cell, it will pack thousands of mitochondria inside to keep the power flowing. But what's fascinating here is what happens when you look closely at the genetic makeup of a mitochondrion.
What do you mean?
Well, they don't just follow orders from the main nucleus. They actually possess their own completely separate circular loop of DNA.
Wait, I thought all our genetic code was locked in the nuclear vault. You're saying mitochondria have a totally separate instruction manual.
They do. They can even divide and multiply independently of the rest of the cell.
That is wild.
And it gets weirder. Earlier, you mentioned ribosomes, the little molecular machines that build proteins. Human eukaryotic cells use a very specific, large type of ribosome, classified as an 80S ribosome.
The S stands for Svedberg units, right? Based on how fast they sink to the bottom of a test tube when spun in a centrifuge.
Exactly. So eukaryotic cells use the larger 80S ribosomes. But inside the mitochondria, they use a smaller version, the 70S ribosome. And that specific 70S size is otherwise only found in simple single-celled bacteria.
Wait, hold on. Let me make sure I'm following this. The power plants inside our cells have separate non-human DNA, and they manufacture proteins using bacterial machinery. Are mitochondria just like ancient bacteria living inside us?
You've hit squarely on the endosymbiotic theory, yes. The scientific consensus is that millions of years ago, mitochondria were free-living, independent eubacteria. At some point, an early eukaryotic cell engulfed one of these bacteria, but instead of digesting it, they formed a symbiotic relationship.
Oh, so they work together.
Exactly. The bacteria had a safe place to live, and the larger cell got a massive boost in energy production. Over eons, they became permanently integrated into our cellular architecture.
We are powered by domesticated microscopic aliens. That is incredible. But, okay, if the nucleus is the blueprint room and the domesticated bacteria are running the power grid, how do we actually manufacture things? Because cells have to produce enzymes, hormones, structural proteins.
Let's trace the operational pathway. When the nucleus sends out an order to build a protein, that message travels to the endoplasmic reticulum, or ER.
Right.
It's a vast 3D labyrinth of membrane-bound tunnels spreading outward from the nucleus. Now, the rough endoplasmic reticulum, the RER, is studded on the outside with those large ADS ribosomes.
Hence the rough appearance under the electron microscope.
Right, exactly. Those ribosomes read the genetic code and synthesize raw proteins, like digestive enzymes. The RER acts as the factory floor, isolating these newly built proteins and transporting them.
But there's also a smooth endoplasmic reticulum, which has no ribosomes at all. What is that doing if it's not making proteins?
The smooth ER is the chemical synthesis lab for non-proteins. It manufactures lipids and steroids.
Oh.
For instance, the specialized cells in human tests that produce the steroid hormone testosterone. Their cytoplasm is absolutely packed with smooth ER.
Okay, so the factory has produced a raw protein or a raw lipid, but usually these things need to be modified or shipped to a specific location in the body. They don't just float away, do they?
No, and that brings us to the Golgi apparatus, discovered in 1898 by Camillo Golgi. Under the microscope, it looks like a stack of flattened, hollow pancakes.
Okay, pancakes.
Yeah, and the raw proteins arrive from the rough ER, and the Golgi apparatus acts as the finishing and shipping department. It might take a simple protein and attach carbohydrate molecules to it, basically transforming it into sticky mucus.
And once it's finished,
The Golgi pinches off a tiny piece of its own membrane to form a biological shipping box called a vesicle. This vesicle travels to the very edge of the cell, merges seamlessly with the outer cell membrane, and pushes its contents out into the body. That process is called exocytosis.
it is a brilliantly coordinated assembly line. It's like an Amazon fulfillment center adding shipping labels. But no factory is perfect, right? Machinery breaks down, proteins get misfolded, DNA gets mutated. How does the cell handle quality control?
By utilizing a specialized bomb squad called lysosomes. These are dark spherical organelles filled with incredibly potent destructive digestive enzymes.
Oh, wow.
They patrol the cytoplasm. If an organelle wears out, the lysosome basically consumes it and breaks it down for parts.
But they have a much more dramatic function, right? Apoptosis.
Yes. If a cell sustains severe DNA damage, or becomes infected by a virus, the cell initiates programmed cell death, which is apoptosis. The lysosomes intentionally rupture, releasing all their corrosive enzymes at once, effectively digesting the entire cell from the inside out so it doesn't harm the wider organism.
It's literally an intentional self-destruct sequence.
It is.
And the balance of that process is critical. If apoptosis happens too aggressively, you end up with massive tissue damage. I know that's the mechanism behind a lot of autoimmune diseases like rheumatoid arthritis, where the body cells start destroying joint cartilage unnecessarily.
Exactly. Or consider the opposite extreme, failed apoptosis. If a cell's DNA mutates, but the self-destruct sequence is broken, that rogue cell just keeps dividing, ignoring all chemical stop signs.
Right.
That uncontrolled immortal division is the fundamental mechanism of cancer. But, apoptosis isn't always about damage control. It's also a vital architectural tool. In the womb, human fetuses actually develop webbing between their fingers and toes. Carefully timed apoptosis destroys those specific cells, sculpting out individual digits.
That's amazing. So our eukaryotic cells are these wildly complex, heavily armed, highly regulated fortresses. But we don't live in a vacuum. We share this planet with prokaryotic cells, simple single-celled organisms, bacteria.
We certainly do.
And here's where it gets really interesting. Because if our cells have literal bomb squads, how does a simple bacterium ever manage to breach our defenses and make us sick?
Well, because bacteria play by a completely different set of evolutionary rules. Prokaryotes don't bother with a membrane-bound nucleus. Their entire genetic code is just a single, naked, circular loop of DNA floating freely in a region called the nucleoid.
Right, they don't have all the complex organelles.
No, they don't have an ER or a Golgi or mitochondria.
But they do have those smaller 70S ribosomes floating around to synthesize their own proteins. And while they might lack internal compartments, their external armor is pretty formidable.
Very formidable. Almost all bacteria have a rigid cell wall made of a polymer called peptidoglycan. This wall prevents the bacterium from swelling up and bursting and gives it a distinct shape.
Okay.
On top of that wall, many disease-causing bacteria secrete a thick, slimy capsule. This capsule acts like an invisibility cloak, hiding the bacteria's chemical markers so our white blood cells can't easily detect them.
Plus, they are highly mobile. Many feature flagella, which are these long, corkscrew-like protein threads that act as microscopic outboard motors. They can spin at 100 revolutions per second, propelling the bacteria through fluid.
Yeah, they're incredibly fast for their size.
They also have pili, right? These tiny hair-like projections covering their surface. They use pilia to physically anchor themselves to our human cells or to attach to each other.
They do. Though pili are a structural vulnerability too. Specific viruses that hunt bacteria, known as bacteriophages, often use the pili as a docking station to inject their deadly viral DNA.
Oh, interesting.
But you mentioned bacteria attaching to each other, and that brings up arguably their most dangerous biological weapon, plasmids.
I am so fascinated by plasmids. Walk us through how these work.
So alongside their main loop of DNA, bacteria often carry tiny secondary circles of genetic material called plasmids. These plasmids don't contain the core instructions for life. Instead, they carry specific, highly advantageous cheat codes, like the instructions for antibiotic resistance.
Okay.
When two bacteria connect using their pili, they can actually replicate a plasmid and pass it across to their neighbor.
Wait, so they are actively trading survival codes on the battlefield? Like a completely harmless bacterium could bump into a dangerous one and just download the genetic software for antibiotic resistance.
Exactly. It's called horizontal gene transfer, and it is a massive headache for modern medicine. Because bacteria also reproduce exceptionally quickly, a single resistant plasmid can spread through an entire population in hours.
So if a patient shows up with a severe bacterial infection, doctors need to know exactly what kind of armor they are dealing with. How do we classify them?
First, by shape. Under a microscope, you'll see spheres called cocci, rods called bacilli, comma shapes called vibrios, and twisted spirals called spirilla.
Okay, shape makes sense.
You also classify them by how they breathe. Obligate aerobes require oxygen. Facultative anaerobes prefer oxygen but can survive without it. And obligate anaerobes are literally poisoned by oxygen gas.
But the most important diagnostic tool has to be Gram staining, right? Developed in 1884 by Christian Gram. I know it divides bacteria into two camps based on color. Gram-positive turns purple and Gram-negative turns red.
That's right.
But why does a doctor care what color bacteria turn in a lab? What is the actual chemical mechanism there?
Well, it all comes back to the architectural differences in that peptidoglycan cell wall we talked about. In the Gram stain process, you first flood the bacteria with a crystal violet dye, followed by iodine. Then you wash the slide with alcohol,
Why the alcohol wash?
because the alcohol is a solvent. Gram-positive bacteria have a massively thick layer of peptidoglycan, heavily laced with a compound called teichoic acid. This thick, acidic net traps the violet dye so tightly that the alcohol wash basically can't pull it out.
Oh, I see.
So gram-positive bacteria stay a deep, dark purple.
Ah, and gram-negative bacteria.
Gram-negative bacteria have a much thinner peptidoglycan layer and zero teichoic acid. Furthermore, they have a whole extra outer membrane made of fatty lipopolysaccharides.
Okay, so when you add the alcohol,
When you hit them with the alcohol wash, the alcohol dissolves that outer fat layer, breaches the thin wall, and completely flushes out the purple dye. When you follow up with a red counterstain, the now transparent gram-negative bacteria absorb it and turn pinkish red.
so a doctor looking through a microscope can immediately see the structural weakness of the invader. If it's purple, it has a thick exposed peptidoglycan wall.
And that tells the doctor precisely which weapon to deploy. Beta-lactam antibiotics, like penicillin, work by specifically blocking the enzymes that build peptidoglycan.
Oh, brilliant.
Yeah, so if a gram-positive bacterium tries to divide while penicillin is present, it can't build a new wall. The internal pressure builds up and their bacterium ruptures.
But penicillin would be basically useless against a red gram-negative infection because their thin peptidoglycan layer is hidden safely beneath that outer fat membrane.
Exactly. For a gram-negative infection, a doctor would pivot to polypeptide antibiotics, like polymyxins, which are chemically designed to dissolve that specific outer lipid membrane.
Wow.
And the absolute brilliance of this medical strategy is the target selection. Human cells do not possess peptidoglycan walls. We don't have those lipid outer capsules.
So we can just flood the body with penicillin, and it acts like a smart bomb. It chemically obliterates the structural integrity of the bacteria while bouncing harmlessly off our human cells.
Precisely.
Okay, we have drilled all the way down into the chemical warfare happening at the cellular membrane. Let's zoom out. How do we get from a single specialized cell to a complex macroorganism like the listener?
Through rigorous cooperation. A single cell alone is incredibly limited. But when identical specialized cells group together to perform a collective function, they form a tissue.
Like epithelial tissues, which are essentially the body's wallpaper. You have incredibly thin squamous epithelium lining the inside of your lungs, specifically designed to be thin enough for oxygen to pass through into your blood.
Right.
Meanwhile, your windpipe is lined with ciliated epithelium cells covered in waving hairs interlocking with goblet cells that pump out mucus to trap inhaled dust.
Yeah, and you also have connective tissues, which provide the structural scaffolding. Bone, cartilage, and the packing tissue around your internal organs. Add in muscle tissue for movement and nervous tissue for electrical communication, and you basically have the basic fabric of life.
I always think of this hierarchy like a developing human society.
Oh, how so?
Well, a single free-living cell is a solo freelancer. But eventually, the freelancers realize they are more efficient if they group into specialized guilds. That's your tissues, the plumbers union, the electricians.
Okay.
But a tissue alone can't build a house. The guilds have to collaborate to form a company. That company is an organ.
That's a great way to visualize it. The heart is an organ made of muscle tissue, nervous tissue, and connective tissue all working in concert.
Exactly.
Even plants rely on organs. A single leaf is a complex organ. It has an outer epidermis for protection, internal mesophyll tissue packed with chloroplasts to perform photosynthesis, and vascular tissue, the xylem and phloem acting as plumbing to transport water and sugars.
And finally, the companies work together to form the global economy. Organ systems. The stomach, intestines, and liver forming the digestive system.
Yeah. And if we connect this to the bigger picture, it is this exact, unbroken chain of complexity that makes life possible. From the raw DNA blueprint to the manufacturing ER to the cooperative tissues and the synchronized organ systems, it all scales up to create a thinking, breathing human being.
So what does this all mean? We started our journey with the immortal HeLa cells of Henrietta Lacks, an anomaly that basically blew the doors off medical research. We learned how researchers bypass the limits of light using electrons in a vacuum to map the organelles of our cellular metropolis.
We did.
We discovered the domesticated alien bacteria powering our muscles, and we unpacked the brilliant chemical logic behind the gram stain and targeted antibiotics.
But if there was one mechanism from our sources that demands a final thought, it is the bacteria's use of plasmids. We discussed how quickly bacteria reproduce and how easily they can trade genetic codes for antibiotic resistance across their pili.
The ultimate horizontal gene transfer.
Right. And while our antibiotics are incredibly clever at targeting bacterial walls, the bacteria are constantly iterating their defenses. It raises a sobering question, really. Are we locked in an unwinnable, accelerating microscopic arms race?
That's a good question.
Yeah, as the bacteria inside and around us continually trade these resistance plasmids, could they eventually outpace our chemical smart bombs entirely?
It's a slightly terrifying thought, but an important one. Something to ponder the next time you get a simple paper cut and your invisible microscopic bomb squads go to war for you. Thanks for joining us on this deep dive into the hidden universe inside you. Keep questioning the invisible world around you.